the cell keeps the virus. I'm always saying we or you because I think of the human body as the host, or the tissue in the human body as the host. But basically, the virus is contained in the cells, and it's not immediately lysed. So the cell stays intact, can live with the virus, and that can go on for weeks. It can go on for a lifetime, and it can come to these outbreaks. For example, measles virus is one of them, where you have those viruses in brain cells, and they stay there for a long time, and you can have outbreaks years later, and they damage a lot of. Yes.
Would that be like the cold sore type of virus?
Oh, we just talked about that.
Those are persistent.
Yeah, because probably one of them.
You never really get rid of them. Right?
Yeah.
But what about something like HPV that can last for 8 years and then shed it completely?
And then what was?
They say you can shed it completely, so they stick around for a while until. I mean, is it in cells to cells replace themselves?
And that's without medication, or?
Yeah.
It really depends on maybe there's almost like something like a critical mass where you have enough viruses, or you have it. Like I said, there's signals by your body, for example, signals that create outbreaks, or that create the replication and the lysis, et cetera, et cetera. How you end up eventually getting rid of it, I mean, it's probably a more uncommon case. I don't know how that specifically works, but I would say, your immune system is probably at some point absolutely adapted to that specific structure. So it could be that you have an outbreak, and then it can kill most of it, or I don't know how that specific case would work. But in general, the persistent infections just mean the host cell can live with it without being lysed, and the consequences aren't cell death, which would then create a lot more of a problem than just keeping the viral information in your cell. And so I think this is for cancer, we would call that a transformation, because the genetic material of the host cell is altered. And so I already covered this really. You have increased the transformation can lead to an increase of growth, and the division of cells is accelerated and is not controlled, which can then result in tumors. And what they call them is oncoviruses. I don't know if you've heard of oncogenes, but oncogenes would be genes that are involved in cancers, and so oncoviruses really are some that are connected to the creation, or to connected to tumors. I don't know if they're, to what extent the virus itself is responsible. So now going to the bacteriophages, you looked at the multiplication cycle, and remember how they attach to the cell, and how the cell is able to enter through fusion or endocytosis, and how it leads through exocytosis. And in bacteriophages, it looks a little bit different. The viruses are more complex, and it's the spaceship design, which means there's a little bit more to it than you've seen in the naked ones, or in the enveloped ones. And what really happens is, once they dock onto one of those prokaryotic cells, they really only they're able to get the DNA, or the RNA, whatever nucleic acid they have, into the cell without going in completely. So they're basically docked like a landing ship on a surface of a planet, and all they do is get the DNA or RNA into the prokaryotic cell. So uncoding is not really necessary in this case. And then usually what happens in those cells is once you have the 100, 200 virus particles and the virus is assembled, the cells usually rupture, so you have lysis. So it's called a lytic cycle, because lysis is really what you have as a mechanism of release for prokaryotic cells that are invaded by bacteriophages. So again, attachment of the virus to specific molecules, penetration, and so this time it only is the genome that enters the host cell, so it seems like an easier, a smarter way to get your genome entered, because you really do what you really only need is the genome in there, and it tells you, if you think about it, the whole structure really does nothing but enter the genome, and then the rest is done by the host cell. So the whole rest of the virus can stay outside and wait until offspring is released.
Do they inject multiple cells, or do they?
Not just the nucleic acid, so really just
No, but I mean, does the bacteriophage do one cell?
No, I think
move on and do another one?
it stays on it, and then you're ambitious for. I mean, they're already so. In my mind, they're already so crazy. I'm glad that one only produces 200 offspring in a short amount of time, but you think it should just hop to the next. Actually, once the if you think about it, once the nucleic acid is released, there wouldn't be anything left.
Only got
So
one dose.
so they're done with their multiplication cycle. But it's something they should keep in mind. And then assembly, just the same thing. So really, the differences aren't that big. Maturation, the completion, and then lysis, important. The cells get destroyed, and so this is really the lytic cycle. We start off with the bacteriophage docking on here onto the cell membrane wall, and then you have the chromosome, the DNA here, the circular one of the prokaryotic cell, the injection of the nucleic acid, and then it creates all these pieces, and it creates all these pieces, the tail fibers and I think there's a collar, so it creates all this whole structure, and then it assembles it, and once you have that many assembled viruses or virions, they mature. Putting them completely together, they mature, and you have so many that the weakened cell breaks open and releases the virus. And this is really what usually happens, but you also have a lysogenic state. So this is really where we focus next. This is really similar to what you've seen before, except the virus doesn't have to get into the cell. But there's also this, and we've seen that in a similar fashion with eukaryotic cell, that there's actually a latent phase where the viral DNA, which is here, or RNA in blue, is enclosed or becomes part of the prokaryotic chromosome, and so it just stays in there, and that's called lysogenic state. So it's a latent state. So it's also called a silent virus infection lysogeny. Not all phages complete the lytic cycle, so that lytic cycle is what usually happens. The completion would be release of viruses out of the cell. So there's some phages that absorb, they penetrate, and then they don't replicate. That viral genome just stays into the bacterial genome, and so it becomes an inactive prophage. The cell is not lysed. And what the advantage is, or what you can imagine, if you think you're basically now in a cell and you included your nucleic acid in that cell, so if you would have, probably won't have to do this, but that would be the bacterial genome, and then you have the viral one. So now it's included in the genetic information of the bacteria, and bacteria are also very fast reproducing organisms, and it also reproduces the virus. So it doesn't just reproduce, it basically reproduces the whole genome of the bacteria, and so the viral nucleic acids or genetic information is replicated and passed on to all these next bacterial cells without doing anything. So it just sits there and it becomes part of all these bacteria that come from this one. So it kind of
That's how mutation happens?
No, that's not really, that's not really why mutations happen, or why mutations happen. There's different ways, but it does change. This is not really a mutation, because a mutation would more look like the genome of a species being changed in very distinct places, and this is an inclusion, so it would be a couple bases that change and that maybe make a protein look different or things like that. And it's because the replication doesn't always work out. There's always little mistakes, and sometimes they slip through, and you have a protein that changes, you have a color that changes, things like that. A color as a product of a change. But this is really an inclusion of a whole genome of a virus. And so these bacteria might be more toxic than we talked about at the beginning. And then that's what I was referring to earlier, the induction. So at some point, these prophages that are not really active can be induced, and then they replicate and lead to cell lysis. So basically, the virus is in there. The bacteria does all the copying for the virus, doesn't even need another phage. It just spreads, and then eventually, through some signal, these start being read, produces virus, and all these bacterial cells all of a sudden lyse, or over a short period of time lyse, and the virus gets out. So it's a latent phase where you say, well, it's not active, but it could be active in the future.
And they all go at the same time?
Well, depending on, if it's, depending on the signal, it depends on what initiates, what induces the production. But you would imagine that if it's, because of the size of it's induced by, say, a specific signal, you would imagine a lot of those cells receiving that signal at the same time. And then the replication and lysis is probably going to take about the same time for each cell. So lysogeny results in the spread of the virus without killing the host cell. And as I said, they cause, a lot of times their toxin or enzymes can cause pathology, or can cause increased toxicity, and they call that a lysogenic conversion, or you would call that a lysogenic conversion. So it's converted through this mechanism, and there's Corynebacterium diphtheria is one of them, cholera, Vibrio cholerae, or Clostridium botulinum, Clostridium, and botulinus toxin, I think, is one of the most toxic substances you can find. So it's a bacterial toxin, but it is emphasized or increases in toxicity. And maybe I can look into those, and maybe we can cover a few of them for.
So the botulism is like bacterial plus virus?
Yeah. And that's why we're really interested in bacteriophages, because there's this notion that together the toxicity increases because of those conversions, because of the viral nucleic acid in the bacterial cells. And maybe, like I said, I might be able to cover those bacteria for disease, and we can talk about them a little bit more, and then you can come back and say, oh, it's because of the viral, the part that the virus plays in this. So, if you compare the two, vermifusine, endocytosis, and then the bacteriophages, this is really what it looks like. So you would have like the head, which looks like a regular capsid with the nucleic acid in there, but it really is. It is pushed in. This is the nucleic acid. It's pushed into the cell through a tube. You can all look up what structures there are on the what fibers, and that's the collar. But in general, what you really see is the main difference is there isn't really that much difference in the synthesis and in the assembly. The release is different because you have usually a lysis. Sometimes they keep them in, and in the bacterial or in the animal viruses, you sometimes have lysis, but you also have exocytosis, budding off of the host cell. Here you always have lysis if it's not the other cycle that they go through. And the penetration, like we said, is different. You have just the nucleic acid versus the whole virus. And here you have capsid or envelope receptors, or not receptors, but you have capsid or envelope proteins that attach to receptors. And for the bacteriophage, you have tail fibers that attach to the cell wall of the prokaryote. So there's differences between those two, but in general, all they're trying to get in is their nucleic acid. And this is a picture of one of them, so you can see in this one, I think there's some, empty ones, which would be the ones that actually attached initially to the cell, and then you've got all these cells, basically these these viruses coming out of this cell and ready to attach to the next host cell. So the problem is if you have a species, or not a species, but if you have an organism or a pathogen, a microorganism, since we talk about those all the time here, that only lives in the host cell, how do you actually study it? Because you're always worried about keeping it in host cells. You can't study them. I mean, if you would study them on a sterilized stainless steel plate, you would probably, first of all, have not a lot of time, and you would imagine that anything outside their natural habitat is going to change things. So you never know if that's really, if anything would be visible or possible to see. You wouldn't know if that's what's really happening. So what you really need to do then is you have to create cells, or you have to create a cell culture in which the virus can live. And why would you do that? Like, what's the main interest? I think mostly, so if you have a case coming in, you say, okay, it looks like a viral infection, you want to identify the virus. So you kind of have to look at what do the cells look like that are infected, what do the particles look like, things like that. And then you could use electron microscopy. For vaccine production, which basically means you have a cell produce vaccines, or you have a cell produce particles for you that then can be injected into, say, an animal, and then the animal produces an immune response to those which are not pathogenic or are not creating the problem. So you want initiating the disease, so you want to produce vaccines. That's what you need those cells for, and you also want to just know more about viruses in general. So that's why you a lot of times try to cultivate viruses and be able to identify them. And what you can do is either you have, and it's over here, you have, I'll go back to the last slide in a second, you have cell cultures, which, ideally they're like single layers of cells, and they grow in sheets, and they basically are the media that we use. If you use nutrient agar or some kind of media for bacterial growth, you use cells because that's what the virus is replicating in, and that's where the virus is multiplying in. And you can look at the effects of them in those cell cultures. And then another really important place to look at viruses to the effects of virus, or at the effects of viruses, but also to produce vaccines, for example, is the bird embryo or a bird egg, because you have a lot of nutrients in there, you have a lot of different tissues in there, and it's all covered with a shell that won't let fungi enter or other bacteria. So it's a perfect, it's really the perfect medium for growth of bacteria, and then you can. You have to be super sterile when you inject those eggs, but it's a perfect system. It's a single-celled system basically to look at virus to look at viral multiplication. And then also what you can find is in some cases live animal inoculation when necessary. It's obviously something that's debatable, but there's a big center at UC Davis, the primate center, where I'm sure some of that has to happen because in the end we're always most worried about ourselves, so we're going to sacrifice anything, at least anything we can contain without without getting too much trouble. Huh?
Like other humans?
Yeah. That's really is the biggest part. I mean, I've done some human studies that is just nutrition based. They take blood from you. They want to know how fast do you, they give you, say, some super slightly radioactive material to see how you digest or whatever. It's totally fine, but I would never want to run a human subject study. It's unbelievable what you have to go through to get those approved, but at the same time that's a good thing, I guess.
Well, compared to what they used to do, right?
Are you referring to the Germans again? I was born after 1939. No, I do. I do sometimes think about that too. I know. I mean, it's happening wherever people. Yeah, I don't know what's happening. It's still happening. You see what you're saying?
Chernobyl, right?
Well, that was.
That was radioactive.
It was an accident.
Sure.
Disturbing images in my head right now. If you ever visit, you have to go to like some of those places, and they explain pretty well what doctors did, and it's pretty rough time, that's for sure. So these, I already explained those to you. Let's wrap up the viruses. So these are the single-celled cultures that you can observe in and what you usually look for is like how does the viral. So if you have cells, if you basically, it's almost like what you looked at in the laboratory with antibiotics the other day. You would have a virus you would have a cell completely filled with viruses. It lyses, and then you would have a little spot that's clear, and then the virus would start spreading out into the neighboring cells, kill those, spread until you would have these spreading growth of nothing, because the cells that are once formed as a continuous layer are now eaten up by or lysed by the virus. And this is just how. Sorry?
That something together too, would that be a sign?
Yeah, I mean, that could be, one of those,
They'll round up.
yeah, it's hard to say. I mean, that's where you really go into detail about specific viruses and what they do. One other thing is, you have an egg and what they do is there's different, you have different areas where you say, there's different, the yolk sac, and there's different areas. So depending on what they're looking for, depending on where you think you're going to get the best growth or the most important results, you can actually inject in different areas of the, like say a chicken egg. So it's pretty interesting what they're able to do. I always wonder about those. I mean, influenza, wasn't it last winter that a lot of people got even though they got vaccinated? And, they said there wasn't a lot of vaccine around anyway, so I'm always imagining these people injecting unbelievable amounts of eggs and, producing that vaccine. But yeah.
Is it all grown in eggs?
I don't know. To look that up too. It's going to show up at the next slide.
And how do they tell where they are if the shell is on it? How can they?
Well, there's I'm sure there's techniques involved, and there's I mean, it's still a pretty big I guess organism or organism to work with. It's they've done much more much smaller, or there's a lot of things we've done that require a lot less material. So it's I'm sure they have specific techniques. I don't know if for vaccine production, for example, if you have to actually go into a specific space, if it's just basically a production of parts in the whole egg, so it doesn't have to be in a specific area. So, and that kind of closes the cycle or circle, the cycle. We're really interested in them because of how they affect us, because they're the most common cause of acute infections. We see them here, we see them in other, in many other, or in all of other countries. They sometimes have high mortality rates. So, a lot of times we realize that for specific viruses, the mortality rate is so high that we really have to figure out how to deal with them. There's a lot of connections we don't know yet between viruses and other, certain cancers, certain disease. Same for prokaryotic or bacteria, where we don't know how much of our chronic disease have their origin or their induction by those. So we're still studying that all the time. And they're obviously also major participants in the Earth ecosystem. So all the ecosystems contain viruses, and they have functions which we're not that concerned with here. And then for the last couple slides, what they do then is if you have an infection, or if you're interested in a specific virus, it's pretty hard to deal with because you need a magnification that a regular microscope or better microscopes won't get, and you have to look at the overall clinical picture. That's one of the ways to go. So you look at what are the symptoms first, and then you take a sample, and then you may infect a cell culture and look at what does the virus do to the cells? What do the cells look like? What do the are there enclosures? What's going on? And then you would look at you could look at parts of the virus. You could look at the response of the immune system to the virus. So you look at what antibodies have formed, and it's hard to deal with because antiviral drugs they really usually affect the host cell function. So you basically try to make sure that virus is not multiplying fast or multiplying at all by preventing the host cell to produce that. So it has a lot of side effects, and that's why it's always a little bit difficult to work with those in comparison to other pathogens. And a couple that I want to just mention because they've been in the in the debate. There's even smaller ones. So if I tell you that viruses aren't even, maybe not even living organisms because they don't have even a cell structure and they're so basic. But there's something that's called prions, and they're basically just protein molecules, and they're misfolded. They don't even contain nucleic acid, and they're also unfortunately extremely resistant to usual sterilization techniques. And you find them, they might be a leftover through evolutionary progress, but you can find them in cell membranes of plants, yeast, animals. But what they sometimes do is because they're an abnormal version of a protein, they might also result in abnormal folding of normal or natural proteins, and that can damage. If you think of like a cell producing a protein, and it's producing protein. Remember the different protein structures, how they're folded. You have a primary, secondary, tertiary, and quaternary structure, and if you think of how they're folded, and if they're folded wrong, there could be a lot of there are a lot of problems associated with that. So it could damage and kill the cell, and they're really the main cause for spongiform encephalopathies. So they have to do with those prions are a cause for a change of nerve cells, and that can cause a lot of problems, and also, which is the same thing. So neurodegenerative diseases, and I'll get to those, and then you know what we're talking about. So connected to prions are diseases like scrapie or BSE, mad cow disease, where you just where those prion fibers are attached, I guess, to the brain cells, and they just cause the neurons to change their structure, and that's why they call it spongiform over here, spongiform. And there's one inhaled wasting disease, and then the Creutzfeldt-Jakob syndrome, and that was pretty big, or it was a big concern for a while, because they also thought these prions would be very specific, but then all of a sudden it was possible to actually get these to have prions from an animal, a species like the cow, affect us. And so that was, I think it was, there's no case in the U.S., but there was a pretty big problem, I think in the UK and mostly. And I don't know if there were any, so I think in the U.S. they had found some of those prions in cows, but never in humans. But in Europe, it seemed to have spread from cows through consumption of beef to humans, and that's always a huge concern. And then lastly, and just know that, so you've heard them. There's satellite viruses. To make it more complicated, they depend on other viruses for replication. So they're really they're only able to live in or replicate in cells where already you have a virus in it. And then one other thing, and just so you heard it, don't try to memorize it. It's viroids. They're only identified in plants, and they're short pieces of RNA. So for all those, you end up for prion, satellite viruses, and viroids, you end up having even less than viruses contain. Not even. It's basically just nucleic acids. There's not even, or in prions it's a protein, but there's not even any other structure. So they're just pathogens or microorganisms we deal with that have really nothing. They're absolutely at the bottom in terms of complexity and their overall structure. Okay, let's have a break till 10:45.
And then we'll